Hello everyone. Today we are going to start with chapter 8, which is cell, the unit of life. In this world, there are two types of things, as we all know, living things and non-living things, which are in living things where the cell is present, in non-living things the cell is absent. So these are inanimate things. Okay. Coming to the living organisms, where the basic structural and functional unit of life, the cell, is present. There are two types based on presence of number of cells in an organism. These are unicellular organisms and multicellular organisms. As we all know, in unicellular organisms, single cell is present. So these are also known as single celled organisms, which are capable of independent existence and performing all its necessary functions of life within one cell. Where here independent existence mean they can survive performing all its essential life functions like metabolism, reproduction, response to any external stimuli without relying on any other cells. Examples of this can be bacteria, which is E. coli and lactobacillus species, protists, amoeba, in fungi, yeast, and in algae, chlamydomonas species, and etc. Coming to multicellular organisms, defined as living organisms which are composed of more than one cell, which have a specialized cells which perform different specialized functions. Okay, and here these multicellular organisms which have complex structure, where cells are organized into tissues, which in turn organize into organs, which in turn organize into organ systems which perform various specialized functions in our body. Examples of multicellular organisms: humans, plants, in fungi, mushrooms, molds, etc., in algae, seaweeds, red algae, kelps, which are known grasses, etc. Note, anything whatever may be, which is less than having a complete structure of cell, does not ensure independent living. Coming to the scientists who described a living cell and nucleus, there are two scientists. Coming to first one, Anton van Leeuwenhoek, who first seen and described a living cell under the microscope. He is known as a father of microbiology. He improved microscope designs and discovered many microorganisms. He observed the living cell using his handcrafted microscope in 1670s. And he also described many microorganisms like bacteria, Streptococcus and Staphylococcus species, which are arranged in clusters like grape-like clusters and chains. In Protozoa, RPC, sperm cells, etc. Coming to Robert Brown, who is Scottish botanist, made a significant contributions in botany and cell biology. He discovered a nucleus in 1831 in plant cells. He also described the Brownian motion, and he worked on plant taxonomy and described many new species. Cell theory. Here, three scientists who given cell theory a final shape. So first we are going to discuss about Schleden, who given in 1838. He is German botanist, examined a large number of plants and observed that plants are composed of different kinds of cells which form tissues of plants and perform various functions like photosynthesis, etc. in the plant cells. Coming to Schwann, in 1839, he is British zoologist who studied on different types of animal cells and reported that cells had thin outer protective layer called plasma membrane which is semi-permeable in nature, and concluded that he studied on plant tissues and he found a unique character on the plant cell wall which form an outer protective covering. And on his hypothesis, he proposed that bodies of animals and plants are composed of different kinds of cells and products of cells. Skeleton and Squan together formulated cell theory as all living organisms are composed of cells and product of cells. But they have a drawback that they didn't explain as how new cells were formed from pre-existing cells. Now, Rudolf Virchow in 1855 said that new cells were formed from pre-existing cells. He given a phrase called omni cellulae e cellulae. It's a Latin phrase which means every cell from a pre-existing cell, which is contrast to theory of spontaneous generation, where they explain that the cell arises as such. But here he modified and he explained that every cell from a pre-existing cell. He modified the hypothesis of Skeleton and Squan and given cell theory a final shape. As today we understand, cell theory is that all living organisms are composed of cells and product of cells, which is formulated together by Skeleton and Squan, and all cells arise from pre-existing cells, which is given by Rudolf Virchow. We all know that, as we discussed in our previous video, cell, which is the basic unit of life, there are two types of cells, where animal cell and plant cell. Okay, in cell there is outer protective structure which is cell membrane in animal cell. In plant cell we have unique character which is a cell wall is present, which is an outer protective covering. Within it, a cell membrane is present. So, next, which in turn contains nucleus inside the cell, which is a dense membrane-bound structure, which in turn contains chromosomes, which in turn contains genetic material and DNA. These cells are divided into two types, eukaryotic cell and prokaryotic cell, based on presence of true nucleus in it. Coming to eukaryotic cell, it is defined as with the cells which have a true nucleus, which are membrane-bound nucleus and contain membrane-bound organelles in it. Examples of this: animal cell, plant cell, etc. It contains a membrane-bound organelles called endoplasmic reticulum, Golgi complex, vacuoles, lysosomes, mitochondria, and microbodies. In eukaryotic cell, which mean in here, which is named as eukaryotic because here eu means true, karyotic means nucleus, where it is known as true, contains true nucleus with membrane bound and contain membrane bound organelles. And here it contains non-membrane bound organelles known as centrosome, which helps in cell division. Coming to prokaryotic cells, pro means before, karyotic means nucleus, which is defined as the cells which lacking true nucleus, which mean membrane bound nucleus and membrane bound organelles. Examples of prokaryotic cells are bacteria, viruses, etc. But in this, both contains cytoplasm, which is a semi-fluid matrix which occupies all the volume of the cell, and this is the main area which perform cellular activities of both the plants and animals. and various chemical reactions take place in the cytoplasm. Coming to ribosomes, which are present in both plant, eukaryotic and prokaryotic cells. Ribosomes, which are non-membrane bound organelles, present not only in the cytoplasm, but these are also present in the chloroplast in plants, mitochondria and rough endoplasmic reticulum. Here cell is differentiated based on different sizes, shapes and their activities. In this video, I am going to explain in brief with examples of different sizes of cells present in the organisms. In next video, I am going to explain in detail about the different types of cells and different shapes of the cells. Coming to here, mycoplasma, which has a smallest cell of 0.3 micrometer in length, and bacteria, which has a cell of 3 to 5 micrometers, egg of an ostrich, which is a single largest isolated cell, and human RBC in multicellular organisms, which is 7 micrometer in diameter, in nerve cells, which are longest cells. Coming to different shapes of cells here, which may be disc-like, polygonal, columnar, cuboidal, thread-like and irregular shapes of the cells are present in the organisms. Hello everyone, as we already discussed about prokaryotic cell in brief, in this video we are going to cover it in detail. As we all know, these lack true nucleus and membrane bound organelles. These are smaller cells and multiply more rapidly than the eukaryotic cells. Even though they vary greatly in shape and size, it has a similar cellular organization. These are some of the examples of prokaryotes like bacteria, blue-green algae, mycoplasma, pleuropneumonia-like organisms. As we all know, these are smallest organisms and lack cell wall, which are pathogenic to human beings and animals. Example to this is Mycoplasma pneumonia, which causes atypical pneumonia, which is an abnormal one and requires special treatment for its cure. Here they given 4 basic shapes of bacteria. Let's discuss it in detail. Bacillus, which is rod-shaped bacteria like Mycobacterium tuberculosis, which appears pink colored long slender bacteria and causes tuberculosis and affects majorly lungs. Coccus bacteria, which is spherical in shape like Staphylococcus, which are arranged in grape-like clusters and appears purple violet color while Gram-Stein procedure and causes infections like skin allergies and hospital-acquired infections. Streptococcus, which are arranged in chains, and these are also gram-positive bacteria, causes infections like tonsillitis, pharyngitis, or sore throat as major infection. Vibrio, which is comma-shaped bacteria and causes infection cholera, which has rice watery stool as major symptom. Spirillum, which is spiral-shaped bacteria. Example to this is Spirochates bacteria, which causes syphilis and shows spiral or rotational movements while seen under dark field microscopy in laboratory. Let's discuss cell structure of prokaryote. It contains cell wall which surrounds the plasma membrane, which is selectively permeable in nature, so it interacts with the outside world. It contains fluid matrix which fills entire volume of the cell is known as cytoplasm. It contains nucleus which is not well defined, and the genetic material of the prokaryotic cell is naked because it is not enveloped by any nuclear membrane. It also contains genomic DNA, which is single circular DNA. Outside to the genomic DNA, it contains small circular DNA, which is known as plasmid DNA, which confers unique phenotypic character to the bacteria like resistant against antibiotics, etc. The plasmid DNA, which is mainly used to monitor bacterial DNA transformation, it is the one of the mechanism which is used in horizontal gene transfer. Here DNA transformation is the process where the cell takes free DNA molecule from other bacterial cell and incorporates into its own genome. It is mainly used in genetic engineering and while doing gene therapy. The prokaryotic cell also has inclusion bodies, which are unique forms which are present only in prokaryotes, and it also contains mesosomes. These are specialized differentiated forms and infoldings of the cell membrane. These are characteristic feature of prokaryotes. Here, none of the organelles which are found in prokaryotes are present in eukaryotes except the non-membranous organelles like ribosomes. Let's discuss cell envelope and its modifications. Bacterial cell composed of three-layered complex cell envelope which consists of glycocalyx followed by cell wall and cell membrane. Here, coming to the glycocalyx, which is an outermost layer which is made up of polysaccharide macromolecules differentiated into two types based on its composition and thickness, which forms a loose sheath around the bacteria which is known as slime layer, and if it forms thick or tough capsule, it protects the bacteria from host immune system as it is sticky or gummy in nature. Coming to the cell wall of the bacteria, here the cell wall is a rigid structure which is made up of peptidoglycan layer. This is the reason if we use antibiotics like penicillin group drugs, amoxicillin or ampicillin for some of the bacterial diseases, these antibiotics acts by preventing cross-linking of this peptidoglycan strands causes cell lysis of bacteria, which leads to death of the bacteria and shows bactericidal effects. Here the cell wall functions as it determines the shape of the cell and provides strong structural support to the bacteria by preventing the bacteria from collapsing. The cell wall is different in both gram-positive and gram-negative bacteria. Let's discuss about plasma membrane of the bacteria. As we all know, it is selectively permeable membrane, so it interacts with the outside world. It is structurally similar to that of eukaryotes. Here, mesosomes, which are specialized membranous structures of the plasma membrane, these are formed by extensions of the plasma membrane into the cells and forms in the form of vesicles or tubules, which helps in DNA replication. These mesosomes, which helps in formation of cell wall, DNA replication, as it consists of enzymes, it also helps in secretion processes. It increases the cell surface area of the plasma membrane. Let's discuss about flagella. These are filamentous extensions from the cell wall. This is made up of a special protein known as flagellae. These flagella, which helps in movement of bacteria, it is composed of three parts: filament, hook and basal body. Filament is the longest portion as it extends from surface of the cell to the outside. Let's discuss the flagella with diagram. This is the structure of flagella. It is composed of three parts, that is filament, hook and basal body. These basal body, again it consists of outer rings and inner rings. Filament is the longest portion of flagella where it extends from surface to the outside. Let's discuss the arrangement of these flagella and different bacteria with examples. Here, the flagella is differentiated into four types based on its arrangement. If the flagella is present at both ends, it is known as amphitrichous. If the tuft of flagella is present at one or both ends, it is known as lobotrichous. If the flagella is arranged all around the cell, is known as peritrichous, which we will see in Salmonella typhimurium. Let's discuss about pili, which are elongated tubular structures made up of special protein known as pilin. Even though they do not play a role in movement of the bacteria, it helps in adhesion of the bacteria to the surface of host cells, and it also helps in DNA transfer in while conjugation, which is also known as sex pili. Here, fimbriae, which is small brittle-like fibers which sprouts out of the cell. It also helps the bacteria to attach to the host organisms and rocks in the strings. Let's coming to the fimbriae. These are small brittle-like fibers which sprouts out of the cell, which also helps the bacteria to attach to the host tissues. In some bacteria, it contains membranous extensions which contains pigments in them, which extends into the cytoplasm of bacteria known as chromatophores. Let's discuss about ribosomes and inclusion bodies in the prokaryotic cells. The prokaryotic ribosomes, which are cellular organelles responsible for protein synthesis and translating mRNA into specific sequence of amino acids. These are non-membrane bound organelles which are associated with plasma membrane of the cell. Coming to the size of the ribosomes, which are 17 to 20 nanometers. Here, several ribosomes with single mRNA, it forms polyribosome, which is also known as polysomes, helps in translating mRNA into proteins. Coming to the structure of prokaryotic ribosomes, these are 70S ribosomes which are present together as two subunits. One is larger subunit which is 50S and smaller subunit which is 30S. These are part of 70S ribosome. Here S stands for sweat work unit which measures sedimentation rate. Coming to the inclusion bodies, these are also non-membrane bound organelles which are reserve material in prokaryotes which are stored in cytoplasm of the bacterial cell. Example to this are synophisian granules, phosphate granules and glycogen granules, etc. In some, these are present in case of viral infection because these are the sites of replication which are present as viral inclusion bodies. Example to this is Negri bodies in case of rabies virus which is caused by rhabdovirus. It composed of cytoplasmic proteins. Viral particles are cellular components of the virus. Coming to the gas vacuoles, these are gas-filled structures which helps the prokaryotic bacteria to regulate their position in aquatic environments and provides buoyancy to the prokaryotic bacterial cell which are present in cytoplasm of the bacteria. Hello everyone, as we already covered prokaryotic cell, here we are going to cover about eukaryotic cell in detail. Coming to the etiology of the eukaryotic cell, it is Greek word where eu means true, karyon means nuclear, where it contains true nucleus and membrane bound organelles like endoplasmic reticulum, mitochondria, vacuole, lysosomes, Golgi apparatus, etc. It also contains complex locomotory and cytoskeletal structures which helps in locomotion, and these provide strong structural support to the cells. The genetic material is organized into chromosomes where the nucleus is covered by nuclear membrane. Coming to the examples of the eukaryotic cells, these are examples: protista, fungi, plant and animal cells. As we all know, eukaryotic cells are not identical. Let's differentiate between plant cell and animal cell. Here, plant cell contains cell wall as outermost protective layer, which is made up of cellulose. These plant cell surrounds the plasma membrane, where in animal cell the cell wall is absent. The outermost covering is the plasma membrane. Coming to the size of the cell, plant cell is larger than the animal cell. In mature plant cell, it contains large central vacuole which occupies up to 90% volume of the cell. Coming to the plant cell, animal cell, here the vacuoles are small, many and temporary. Coming to the plant cell, here the plastids are present which involved in photosynthesis. The plastids are absent in the animal cell. The centrioles and centrosomes are present in the animal cell and absent in the plant cell. Let's discuss about structure of plasma membrane, which is also known as cell membrane. As we all know, it is a selectively permeable in nature. It regulates the movement of substances in and out and interacts with the outside world. The structure of plasma membrane is studied on the human red blood cell, and detailed structure of the plasma membrane is studied after the invention of electron microscope in 1950s. Coming to the composition of the plasma membrane, it contains lipids, proteins and carbohydrates. Coming to the lipids, these are 40%, proteins which are 52%, and it also contains carbohydrates on the cell membrane which is very less amount. Coming to the lipids, these also contains phospholipids and cholesterol. The phospholipids which has hydrophilic polar end towards the outside the membrane, which contains saturated hydrocarbons, and it also contains non-volatile towards inner side, which is hydrophobic in nature. Coming to the proteins, on ease of extraction, these are different classified into two types: integral proteins and peripheral proteins. Integral proteins which are partially or totally buried in the membrane. These are also known as transmembrane proteins because these proteins which are present throughout the lipid bilayer. Coming to the peripheral proteins which lie on the surface of the plasma membrane. Carbohydrates, as we already discussed, these are present in very less amount. The most improved model of plasma membrane, which is known as fluid mosaic model, this is proposed by scientists Singer and Nicholson in 1972. According to this model, they said that the lateral movements of proteins within the lipid bilayer is due to quasi-fluid nature of lipids, and this fluid nature of lipids which helps in growth of cell division, endocytosis and secretions, etc. And this ability to move within the membrane is measured as its fluidity. These are the various cell organelles that are present in the eukaryotic cell, like nucleus, lysosome, ribosomes, endoplasmic reticulum, mitochondria, Golgi operators, etc. Let's discuss structure of each and every cell organelle and functions performed by them in detail. Coming to the endomembrane system, it composed of group of membrane-bound organelles like endoplasmic reticulum, Golgi complex, lysosome, vacuoles, which are structurally different but functions in a coordinator manner. The mitochondria, chloroplast, which is present in plants, and peroxisomes, these are not the components of endomembrane system, as these are not functions in a coordinated manner. Coming to the endoplasmic reticulum, these are the tubular structures which are scattered in the cytoplasm. Let's discuss the structure of endoplasmic reticulum in detail with the help of diagram. Here, the endoplasmic reticulum, it divides its intercellular space into two compartments, which are luminal compartment, which is present inside the endoplasmic reticulum, and extraluminal compartment, which is present towards the cytoplasm. Again, based on presence of ribosomes, these endoplasmic reticulum is divides into two types. Those that pierce ribosomes on its surface are known as rough endoplasmic reticulum, which involve involved in the synthesis of proteins, and these rough endoplasmic reticulum which continues with the outer membrane of nucleus. Coming to the smooth endoplasmic reticulum, these do not contain any ribosomes on its surface, and these involved in the synthesis of lipids, and these also synthesized lipid-like steroid hormones. Let's discuss about Golgi operators in detail. These are densely stained reticular structures which are present near the nucleus of the cell. These are first observed by Camillo Golgi in 1898. Coming to the structure of the Golgi operators, it consists of four parts which are cisternae, which has scissor forming phase, and tubules, vesicles, and Golgian vacuoles. Let's discuss structure of each part in detail. Coming to the cisternae, these are flattened sac-like structures which has scissor forming phase and maturing phase. As these two phases different to each other, these are interconnected in its function. These cisternae are parallel to each other and arranged concentrically near the nucleus with diameter 0.5 micrometers to 1 micrometer. Coming to the tubules, these are flattened round structures which arises at the periphery of the cisternae. Vesicles, these are small round structures which arises at the edges of the cisternae. Coming to the Golgian vacuoles, these are large spherical structures of the Golgi operators. Let's discuss functions of the Golgi operators. These helps in processing, packaging, and transporting the materials to the intracellular targets or outside the cell. The proteins which are synthesized in the endoplasmic reticulum are transported towards the cisternae of the Golgi operators and fused to the cis phase of the Golgi operators and moves towards trans phase, a maturing phase of the Golgi operators, where these proteins are modified before they are releasing out of the trans phase, and these are later acts as lysosomes and perform various functions in the cell. Let's discuss the structure of lysosome in detail. These are tiny spherical sac-like membrane-bound vesicular structures which are formed by packing in the Golgi operators. These are rich in hydrolytic enzymes like hydrolysis, which are lipases, carbohydrates, proteases, and nucleases. These are active at acidic pH and helps in capable of digesting carbohydrates, proteins, lipids, and nucleic acids. This also contains transport proteins on the surface of them. Let's discuss about mitochondria. As we all know, this is cylindrical, a sausage-shaped double membrane organelle which is present in the cytoplasm of the cell, which has 0.2 to 1 micrometer in diameter and on an average which is 0.5 micrometers, coming to the length of the mitochondria, which is 1 to 4.1 micrometer in length. And to see the mitochondria under the microscope, we have to use special stain known as Jonas green B. There are variable number of mitochondria are present in the cell, different types of the cells, depending on the physiological activity, shape, size, and energy requirements of the cell. Let's discuss structure of mitochondria in detail with the help of diagram. Coming to the structure, it is double membrane bound organelle which contains two membranes, that is outer membrane which continues as limiting boundary of the mitochondria, and inner membrane which forms number of infoldings known as cristae, and the lumen of mitochondria again divides into two compartments. Outer compartment which is present between the outer and inner membrane of the mitochondria, which is known as intermembrane space or perimitochondrial space. Coming to the inner compartment, which contains dense homogeneous substance known as matrix. These contains circular double-stranded DNA molecules, RNA molecules, ribosomes which are 70S, and these also contains components required for the protein synthesis. Coming to the functions of mitochondria, these are the sites of aerobic respiration, and these are also known as powerhouses of the cells, as these contains enzymes required for the synthesis of ATP, and these produces energy required by the cell to perform various functions. There are two membranes in the mitochondria. The two membranes have its own specific enzymes which are associated with the mitochondrial function. Let's discuss about ribosomes. These are observed by George Pellet in 1953 as dense particles. These are non-membrane bound organelles which are present both in prokaryotic and eukaryotic cell as 70S and 80S ribosomes. It has two subunits, larger subunit and smaller subunit, which is 60S and 40S in the eukaryotic cell. Here in prokaryotic cell, 50S and 30S, this is the larger subunit and this is the smaller subunit. Here S stands for Svedberg unit, which is segmentation coefficient. Here it measures the segmentation rate. Coming to the structure of the ribosomes and its functions, the ribosomes which helps in synthesis of proteins and translating mRNA into specific sequences of amino acids. Let's discuss about cytoskeleton. These are elaborate network of filamentous proteinaceous structures like microtubules, microfilaments, and intermediate filaments which are found in the cytoplasm of the cell and helps in the providing strong structural support to the cell movement of the cell and maintains the shape of the cell. Let's discuss with the help of diagram. These are microtubules, which are hollow cylindrical structures found in the cytoplasm of the cell and made up of tubulin protein, and which forms the cytoskeleton of cilia and flagella. These are microfilaments. These are unbranched structures found in the cytoplasm of the cell and composed of actin and myosin proteins, helps in the cell division by forming an furrow. These are intermediate filaments. These are hollow filamentous structures helps in the forming scaffolds for chromatin material. Let's discuss about structure of centrosomes and centrioles. Here, the centrosome consists of two cylindrical structures which lie perpendicular to each other and has a cartwheel-like organization are known as centrioles. These are surrounded by amorphous pericentriolar materials. Let's discuss structure of centriole in detail. To the structure of centriole, it has 9 evenly spaced peripheral fibrils which are known as triplet fibrils made up of tubulin protein, and these triplets are linked with adjacent triplets with the help of AC linkers, and it also has central proteinaceous hub which are connected to these triplets with the help of radial spokes. These centrioles forms the basal body of cilia and flagella, and spindle fibers gives rise to spindle apparatus during the cell division. Let's discuss cilia and flagella. These are hair-like outgrowths of cell membrane. Coming to the cilia, these are small structures which helps in the movement of cell and surrounding fluid. Here, the flagella, which is longer one, which is responsible for cell movement. Structurally, the prokaryotic flagella is different from the eukaryotic flagella. Here, it consists of basal body, rootlets, basal plate, shaft, and axoneme. Let's discuss it with the help of diagram. Let's see the structure of flagella. It's an axoneme, a shaft of the flagella. It contains 9 microtubules with 2 central tubules which are connected with bridges and enclosed by central sheath. So these flagella has an arrangement of 9 plus 2, where 9 means it has 9 microtubule doublets and 2 means 2 central tubules. And both cilia and flagella emerge from the centriole-like structures called basal bodies. As we already differentiated between plant cell and animal cell, let's discuss some of the organelles which are present only in plant cells like plastids and outermost covering of the plant cell, which is cell wall. Coming to the plastids, these are found in all plant cells and euglenoids. Depending on its color and types of pigments present in them, these are divided into 3 types: leucoplast, chromoplast and chloroplast. Coming to the leucoplast, these are colorless plastids which are present in non-green cells. These are of 3 types: amyloplast, which contains starch, example potato tuber; alleloplast, which stores oils and fats; alluroplast, which stores proteins in them. Coming to the chloroplast, these chromoplasts, which consists of fat-soluble carotenoid pigments like keratin, xanthophyll, etc. These produces colors like yellow, orange or reddish orange to the ripening fruits, because here the chloroplasts are transformed into chromoplasts, which gives colors to the ripening fruit. Coming to the chloroplast, these are lens-shaped oval or spherical ribbon-like structures which are present in the mesophyll cells of the leaves. These are greenish plastids which are present in green cells of the leaves. These contain chlorophyll and carotenoid pigments which are responsible for trapping light energy, which is essential for performing photosynthesis. These are about 5 to 10 micrometer in length, 2 to 4 micrometer in width. There are variable number of chloroplasts are present in cell, like in Chlamydomonas, which are present 1 per cell, and in green algae, these are present 20 to 40 per cell. Let's discuss structure of chloroplast with the help of diagram. Here, the chloroplast is double membrane organelle which consists of 2 membranes, which is outer membrane and inner membrane. Between outer and inner membrane, it consists of space known as intermembrane space. Inside the inner membrane, the fluid-filled space known as stroma, which contains number of flattened membranous sacs known as thylakoids, and these thylakoids are arranged in stack-like piles of coins known as grana, singular, which is known as granum, and these grana are also known as integral thylakoids. And thylakoids of different grana are connected with help of tubules known as stoma lamellae. These consist of 70S ribosomes, which are smaller than the cytoplasmic ribosomes. And these chloroplasts, as we already discussed, these consist of pigments like chlorophyll, which trap light energy essential for photosynthesis. These perform photosynthesis, light reaction in the thylakoids and dark reaction in the stroma. These also contain starch in them. These mitochondria and chloroplasts are semi-autonomous organelles because they are capable of self-duplication, as they have their own nucleic acid like double-stranded DNA and RNA molecules, 70S ribosomes, and it also has ETS and ATP forming machinery in it. Let's discuss about cell wall. It is non-living rigid structure which is found in fungi, algae and plant cell. Here, in fungi, it is composed of chitin. In algae, it is composed of cellulose, galactans, and some of the minerals like calcium carbonate. And in plant cell, it is composed of cellulose, hemicellulose, pectin, and proteins. Based on age of the plant cell, in young plant cell, it forms a single layer which is thin, elastic, and capable of growth, which is known as a primary cell wall. And in mature plant cells, inside to the primary cell wall, it forms as secondary cell wall. Coming to the middle lamella, it is the connecting material which holds the adjacent cells together, as you can see in the picture. Here, the cell wall and middle lamella are transversed by plasmodesmata, which means these plasmodesmata which extends or passes across the cell wall and middle lamella and connects the cytoplasm of neighboring cells, and this allows transport of water, nutrients, and provides cell to cell communication between the plant tissues. Coming to the functions of the cell wall, it maintains shape of cells and protects cell from mechanical damage and infection, and it provides cell to cell interaction, and it provides barrier for undesirable micromolecules. Let's discuss about vacuole. It is present in both plant and animal cells, but in plant cell it occupies large volume up to 90% volume of the cell. Let's discuss structure of vacuole. It is single membrane bound organelle in the cytoplasm. It contains water, sap, excretory products, and waste materials. The single membrane, which is known as tonoplast, it facilitates transport of number of ions and materials against the concentration gradient into the vacuole. There are many types of vacuoles, like contractile vacuole, which is found in amoeba. This helps in osmoregulation and excretion, and food vacuole, which is present in protists, which helps in engulfing the food particles, and gas vacuole, as we already discussed about it in prokaryotic cell. These are found in prokaryotes, which provides buoyancy to the cells. As we already discussed about various cell organelles of eukaryotic cell, its structure and function in detail, here we are going to cover about nucleus, which is the largest cell organelle and controls all the activities of the cell. It was first described by scientist Robert Brown in the year 1831, and based on number of nucleus present in a cell, if the cell contains two nuclei, then it is known as binucleate cell, as in case of paramecium. If cell contains many nuclei, then it is known as multinucleate cell, and if cell lack nucleus, as in case of mature mammalian RPC and sieve-tube cells of vascular plants, then it is known as enucleate cell. Let's discuss about structure of nucleus in detail. At non-dividing phase of the cell, which is known as interphase nucleus, has nucleus chromatin, nuclear membrane, perinuclear space, and nuclear pore. Let's discuss about each and every part of the nucleus in detail. Coming to the nuclear envelope, which is the membrane that separates nucleus from the cytoplasm, is differentiated into outer membrane, which continues with the rough endoplasmic reticulum, and inner membrane, which is smooth. Between these two membranes, there is a space which is known as perinuclear space, and there are nuclear pores which are formed by fission of these two membranes, and this forms as passage of substances to move inside and outside the cell. And there is transparent fluid which fills the nucleus, which is known as nucleoplasm, that contains nucleolus and chromatin material. Coming to the nucleolus, this is non-membranous spherical structure. This is a site for ribosomal RNA synthesis, and there are large number of nucleolus present in a cell, and that indicates that this cell is involved in protein synthesis. If there are large number of nucleolus in a cell, that indicates the cell is actively involved in protein synthesis. Coming to the chromatin material, it's a Greek word where chroma means color. It is a highly extended and elaborate nucleoprotein fibers. Here, it is named by scientist Fleming based on its ability to stain with some of the basic dyes. And this chromatin material, which is composed of DNA molecules, some of the basic proteins like histones, non-histone proteins and RNA molecules. In human cell, the DNA, which is 2 meters long thread-like structure, which is distributed among 23 pairs of chromosomes. At different stages of cell division, where we are going to discuss in chapter 10 cell cycle and cell division in detail, the chromatin material is condensed to form chromosomes. Let's discuss about structure of chromosome in detail. Here, the chromosome consists of 2 identical halves which are known as chromatids, and these chromatids held together at a point which is known as centromere, and this centromere appears as narrow region which is known as primary constriction, and sides of these centromere there is a disc-shaped structures which are known as kinetocodes. Based on position of this centromere, there are 4 types of chromosomes. Let's discuss about each and every type of chromosome in detail. Metacentric chromosomes, if the centromere is present at the center of the chromosome and divides into 2 equal arms. If the centromere is present slightly away from the center of the chromosome, then it divides the chromosome into one shorter and one longer arm. Then it is known as submetacentric chromosome. If the centromere is situated close to its end and divides into one extremely short and one extremely long arm, then it is known as acrocentric chromosome. If the centromere is present at terminal portion, then it appears as single arm. It is known as telocentric chromosome. And few chromosomes has additional constrictions at its end, which is known as secondary constrictions, and which is present beyond to the secondary constrictions as small fragments. These are known as satellites. If you like this video, don't forget to like, share and subscribe to my YouTube channel. We'll meet again in our next video.